So let us quickly go through this thing. So you see this thing, this is the axoneme. So if you're seeing, this is how it will be arranged in a three-dimensional plane. So what you're seeing here is like, this is just a cross section that you're seeing. If you're going to cut it off, you will see a structure like this. So this red, this is the entire thing that you're seeing off. So if you're seeing here, this is the core which is there. Okay, so this entire thing is there. This is the axoneme. So if you're in axoneme, if you're going to cut it and see, you will be able to see something like this. So this diagram you see, you are able to understand. So this is the basal body. This is the entire flagella. So if you're going to see this part alone, so this is the flagella or cilia, whatever it is. If you're going to see that part alone, and if you're going to see here, you will see something like this. Okay, what are the things that you will see? We'll try to study about it now. So you just draw this structure. The structure alone you draw. I'll give you some 2-3 minutes to draw. Quickly draw this structure alone. The circle structure. If you want a simpler version, you can go for this. Okay. Someone is saying something. What is that? Something that they have said. When will the result means? I don't know when the result will come. Okay, so now what we will do is like we'll try to study about it. Let us quickly write some points about axoneme. Centriole structure is quite different, so let us just try to see. Okay, just a second. So what is this axoneme? I haven't started anything explaining at all, so just let us try to study about it first of all. Okay. The transverse section of cilia or flagella shows 9 peripheral doublets and a pair of single central microtubules, forming a 9+2 arrangement. Okay, what is the meaning of this thing? You are able to see that if you are going to take a cilia or flagella in the periphery means in the border. In the border, you will see this kind of doublet. 2, third one, fourth one, fifth one, sixth one, seventh one, eighth one, ninth one. Like this, you will see 9 microtubules in the periphery. So if you're going to cut a cilia in the cross section, if you say that this is the cilia, if you're going to cut it this way, here you will be able to see this thing. So you will be able to see this kind of arrangement. What will you see? All these things are microtubules. But microtubules, are they present singly or are they present as doublet? They are present as doublet. What is the meaning of doublet? Doublet means two things are arranged. Okay. This is there in the 9 peripheral. Peripheral means it is there in the boundary. And a pair of single central. What is the meaning of single central? There is one pair which is there in the single center. Okay. So this kind of arrangement is called as a 9+2. So this is 9 doublet in the periphery. And then in the center, there are 2. This is called as a 9+2 arrangement. So this is what is the first point. So if you are able to see here in the structure, you are able to see the structure. This A and B forms a doublet that is called as a microtubule doublet. Okay. And it is arranged, 9 things are arranged in the periphery and 2 microtubules are arranged in the central region. So the ones which are there arranged in the central region are not doublet. You are able to see that they are only singlets. Singlets means 1 and 1. So this kind of arrangement is called as a 9+2 arrangement. Are we able to understand? So this is the first point. And the second point, this axoneme is covered by a continuous membrane. Okay. So this is kind of it is basically the axoneme that I am trying to show here. So the axoneme is the central part of the cilia. Okay, in cilia and flagella, you will be able to see this thing. Okay, this is covered by a membrane basically. This is the second point. What is actually called as axonema? Yesterday we studied, no. If you are going to talk about a cilia will have cilia or flagella. If you are going to talk about cilia structure or a flagella structure, you will have, if you are talking about a flagella, you will be seeing a structure like this. Okay. If you are talking about a cilia, you will see a structure like this. Okay. So if you are going to talk about it, here it will be attached with the body which is called as the basal body. Okay. And then there is a part which is called as a basal plate. And there is a part which is called as a rootlet. So here, this part is called as the axoneme, or we call it as shaft. This part is called as the basal body. Okay, so now here you have this part, we call this as a basal plate. And these parts are called as the rootlets. Okay, if you are going to talk about cilia also, the cilia will also have the same way. This is what we call it as the axoneme. Here there will be something which is attached, which is called as the basal body. Below that there will be a part which is called as a basal plate. And then there are some structures which will be coming out of it, which we call it as rootlets. So if you are going to talk about cilia or flagella, we are trying to study about the structure of them, how it is organized. That is what we are trying to study. This is how, these are the four parts in a cilia or a flagella. In that particular flagella, you will be able to see some structures which are there inside. What are these things made up of? These are made up of, like inside the thing, microtubules are made. But there is a pattern in which this microtubules are like going through this thing. Or here also there is a pattern in which this microtubule is going. What is the pattern? That is, this is the pattern that you are seeing. You are able to see that there is like kind of a structure. This is like kind of a arranged as a doublet. What is this thing? This is a microtubule. Arranged in the format of 9 plus 2. Axoneme can also be called as shaft. Yes, right. The other name for axoneme is shaft. Are we able to understand? Okay, so now let us move on further. And then you have to write each doublet has a pair of arm that projects from microtubule towards the neighboring doublet. These arms are made up of dynein. It's a protein. So what is that they are trying to tell is, like, you have a doublet. Okay, if you're seeing here, this doublet is there. From here, there is a protein which is trying to connect here. Each of these things, it is like kind of extending towards that thing. Okay, this short arm which is trying to project is called as it is made up of a protein that is called as what? Dynein. Each doublet has a pair of arm. It is not just one arm, it is having a pair of arm it will be projecting from the microtubule towards the neighboring doublet. These arms are made up of what protein? They are made up of a protein which is called as dynein. So if you are able to see here, you are able to see this arms. Two things are like kind of like showing. Each of these two things which are shown here, this is the arm that we are talking about. Okay, so this is one more thing that you need to know. And then there is something called as a radial spoke. Extends from microtubule towards the center. Microtubule. Children, please listen here that I am already like kind of occupied with teaching. I cannot be answering the questions about 10th results. So please try to avoid questions about it. If you're like very much interested in 10th result, you may choose to go out and then check it, withdraw yourself from the class and maybe later see the video. Okay. So if you're here in the class, I would like to focus only on the class. I'm giving you an option. I understand that you are all like kind of anxious about your results and you might want to take an off. If you want, you please go and then later see the video. If you're here inside the class, I want comments related only to the class to me. There is still an option. There is a central sheath covering. There's a second covering the central microtubules. Okay, so I'll just show you in the diagram. Dynein is a protein, that's all. You're able to see here, this line that you're seeing here is the radial spoke. So you're able to see that there is like a kind of a line which is like extending from the doublet to the center. That is called as a radial spoke. And this line that you're seeing here is the central sheath. So you're able to see something like a covering here. So this covering kind of thing is the central sheath. So this is the central sheath and this is what you call it as your radial spoke. Are we guys able to understand? So this is what is the structure. So this is how your what is structured, your axoneme is structured. So with this thing, axoneme is over. Now we'll move on to the basal body. At the end of cilia or flagella, a basal body or kinetosome is found. In the cytoplasm. Okay, and then like the next thing is it is a short cylinder with 9 microtubules triplets in the periphery without central tubules. In 9 plus 0 attachment. Sorry, arrangement. Okay, this is about the basal body. So what is the basal body? You need to know that the basal body is the part which is arranged here. So you will have a basal body which is arranged from which your flagella or the cilia is like kind of like going. Let us say that this is here it is arranged in 9 plus 2 order. But here what you will see here is if you're going to take the basal body and if you're going to see the structure, what you will see is you will see a triplet. Instead of doublet, you will see triplets. Like that you will see triplet kind of arrangement. Okay, like how many? 1, 2, there will be 3, there will be 4, there will be 5, there is 6, there is 7, totally 9. So it is basically 9 triplet will be arranged. Okay, this is like kind of wrong. So if you are going to see here, there will be 1 triplet, second, third, fourth, fifth, sixth, seventh, eighth, ninth. So it will be arranged as a triplet. Three things together. This kind of arrangement is what we call it as a 9. This is the periphery. Periphery means in the outer covering. But there is nothing in the center. So here in the center, nothing is there. So this is called as a 9 plus 0 arrangement. So this is how your basal body will be arranged. You see here, so you are able to see here, this will be arranged like this. Your basal body will be arranged as a triplet. 1, 2, 3, 4, 5, 6, 7, 8, 9. All these things are 9. I mean like 9 things are arranged. What kind of structure is arranged? It is like basal body, basal plate are slightly different structures. They are not the same structures. Tambi, please understand that whatever we are talking here is we are talking about microtubules. Okay, centriole is a different organelle altogether. Cilia is a different organelle altogether. Okay, so here I am seeing people are confusing yourself with the cilia, flagella, okay, and then centriole. We haven't seen centriole at all so far, so please don't get confused with this thing. Cilia, flagella, centriole, all these things are made up of microtubules. Okay, so the microtubules will make cilia, microtubules will make flagella, microtubules will make centriole. So what is microtubule? It is a part which we call it as a cytoskeleton structure. So this is almost like mud. Mud can make like what? Bricks. Mud can make bricks of a different shape. Mud can make bricks of a different consistency altogether. So these are all like kind of different types of bricks. This is mud. Maybe you can understand it that way. Okay. So microtubules are structures that will make cilia. Microtubules are structures that will make flagella. Microtubules are the structures that are going to make centrioles also. Okay, so we haven't come to centriole. Centriole has different role, different organelle it is like next we are going to see into centriole. Okay, as of now what we are studying is we are trying to study about cilia and flagella. Okay, so cilia and flagella are locomotory structures which are made up of microtubules. In the previous class, I wrote flagella has basal plate, basal body, rootlets, and then also the fourth one is axoneme. There are four parts. There are four parts in any of these things. Let us try to see the parts of the flagella. You see here, this is like kind of showing you the structure. So you see here, you're able to see this is the axoneme. You have something called as a basal body. Okay, here you have, it is written as basal foot. This basal foot and the basal plate is the same thing. And then you are having something called as a striated rootlet. This is the rootlet. So all these four are different parts which are found in cilia as well as flagella. We are now seeing, we have studied about two structures. What are the two structures that we have studied about? We have studied about axoneme. Okay, we have studied about the second part, which is the basal body. Then you have something called as a basal plate. That is a third structure. And the fourth structure is your rootlet. So this is the second structure that which was there. So now you have the second structure is over, done. Now we'll try to study about basal plate. In some textbooks, they would give this as what? Basal foot. Okay, so you can just write that thing. In a motile cilia, the boundary between axoneme and the basal body is also known as basal plate. That's all. It's just a single line. Yeah, cilia is motile only. So here you can see the structure. This is the area. This is the boundary. In that area, you will be able to see something called as a basal foot. Okay, this is one thing. And the last thing would be rootlet. The next one is rootlet. They are support structures that arise from basal body. That arise from basal body of cilia. They firmly hold the ciliary structure. Okay, so with this thing, cilia and flagella is over. So I have told you that there are 4 parts here. The basal plate will be more like a boundary. Okay, so you have axoneme or shaft, you have a basal body, you have a basal plate. It is also called as a basal foot, and then you have rootlets. Okay, it will be like rootlets will be like kind of thing. So this is just a covering kind of structure. This basal plate is nothing but a covering kind of structure which is found. Okay, and then like we have studied about what is axoneme. Axoneme has a 9+2 arrangement. Then you have the basal body. The basal body has a 9+0 arrangement. This is a triplet microtubule structure. This is a doublet microtubule structure. Okay, and then you will see that thing what? Then you have studied about basal plate, and then you have studied about rootlet. These are all just associated structure. Basal plate and then like the basal, the rootlet are associate structures. Just like that, they are part of it. The main part of that particular flagella or cilia is only what and what? The axoneme or the shaft, and then the basal body. Those two are the major components. The other things are just support organelles. Are we able to understand? Okay, so with this thing, we are completing what? We are completing the concept of cilia and flagella. So now we'll move on to the next concept. The next concept that we'll be like studying is, we'll be studying about is your centriole. Yeah, flagella is remain attached to the plasma membrane. So you have a cell here. So it will be arranged. The basal body is found in the cytoplasm, and it will be like this. If cilia is there, the basal body is there in the cytoplasm, and then it will be like this, arranged like this. So basically, the basal body is in the cytoplasm in eukaryotic cell, whereas in your prokaryotic cell, the basal body will be there in the plasma membrane itself. Okay, we are now studying about eukaryotic structure, so you concentrate only on the eukaryotic structure. So the prokaryotic flagella and eukaryotic flagella is like kind of different. The next one we are going to see is we are going to study about a centriole. So once centriole is done, it's just the microbodies and nucleus. No need to copy the diagram, you can just check it. Now we can go for centrioles. Or centrosomes. A centrosome is an organelle that contains two cylindrical structures called as centrioles. This is the first point. The second point is that centrioles are submicroscopic barrel shaped structures that occur in the form of two granules. Third point, they are also called as diplosomes. They are located in the cytoplasm. So if you see, excuse me, this is a centrosome. It will have some two cylindrical structures which are lying perpendicular to each other. So these two things are called as the centriole. And this is your centrosome. Okay, so that is what is the first point is trying to tell. The first point is trying to tell that centrosome is an organelle that contains cylindrical structures called as two cylindrical structures called as the centriole. The centrioles are microscopic, barrel shaped. You are able to see that they are looking like a barrel that occur in the form of two granules. So you are having two granules. So we call them as what? Diplosomes. Diplosome means two. Two structures are there. So we call it as diplosome. Where are they located? They are located in the cytoplasm. Okay, so now moving on further, they occur in all plant cells, so they occur in all animal cells. And are absent in in plant cells. Okay. The next thing that you should know is the structure. We'll try to study about the structure. So you have this cell. Inside it is filled with a fluid. This is a centrosome. Inside that there is a fluid. That fluid is called as centrosphere, or we call it as kinoplasm. So in that thing, you will be able to see this barrel-shaped structures. The centrioles are perpendicular. They are in a fluid called kinoplasm, or centrosphere. The cylinder structures are the centriole. The cylinder shaped structure is the centriole. Where is the centriole present? It is present inside a organelle which is called a centrosome, which is filled with the fluid which is called a centrosphere. And where is the centrosome is present? It is centrosome is present in the cell, just like that. Okay, if you're done, you please let me know. So each centriole is made of 9 groups of microtubules. Okay. Each microtubule group is a triplet. They are made up of a protein called tubulin. Okay. The microtubules are arranged in the periphery. Okay, someone is asking for the previous slide for a minute. Yes, copy it and then tell me quickly. Okay, wait a minute. Let me show it in the end. One by one now, everyone has started to ask. Let me just quickly finish it off. There is no central tubule. The arrangement is, what will you call this arrangement as? In the periphery, there are 9 and there is nothing in the central tubule. There is no central tubule. So we call this arrangement as what? It is 9 plus 0 arrangement. Okay, so very good. So good, you guys are all understanding. The adjacent triplets are connected by linker proteins. Okay, so you see here, it is almost like the similar structure that you studied about the basal body. It is almost of the same thing. It is made of a triplet. Like that, triplet and it will be arranged like how many triplets will be arranged? 9, 1, 2, 3, 4, 5, 6, 7, 8, 9. Like that, like 9 triplets will be arranged in the periphery, which is all connected by a linker protein. I'll just quickly show the structure. You see, this is how it will be. You are able to see the two cylindrical structure which are present perpendicular to each other. If you are seeing it in a little closer way, this is how it will be arranged. You are able to see that this is a triplet, 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 this is a triplet. Like this, it is arranged in a circle, and it is all linked by this like blue color structures. Those blue color structures are the what we call it as the linker protein. Okay, you see here a little bit closer view. So you are able to see that this is the ABC. The triplet is what you are showing, seen as ABC. Okay, and then in between you have this linkers which are there. Those linkers are called as the AC linker. Why we call it as a AC linker? Between A and the C. A protein of A microtubule of one and C microtubule of another thing, it will link. So we call this as what? AC linker. Okay, like this it is linking. So we call that as a interlinking protein. And from here, what is happening? You are able to get as spikes or spokes, radial spokes. These radial spokes are all like integrated to form what we call it as a core. Okay, you are able to see a radial spokes that is all connected in the center to form the core. It does not look like a cylinder. You see here, this is what is the cylinder. This is the three-dimensional structure. This is just a cross-sectional figure that you are taking. So if you are going to see only the side, it will look like this. This is not a cylinder. You see here, this is how it will look like. The central microtubules are absent. That is why we call it as what? We call it as a 9+0 arrangement. What, Sharon, you are able to understand the cylinder structure? So this is how it will look like a cylinder, but if you are seeing the side, you will be getting it. I am not sure about the size of it, man. Okay, you can check the internet, and it doesn't. You don't require whether the size is 9 nanometer or 10 nanometer, because it will vary from cell to cell. Then you write 2 more points with which we will complete this thing. The spokes arise from microtubules and connect in the center to the hub. Spokes are proteinaceous strands. Spokes are connected to you have this triplet. Each triplet is there. In between this triplet, there is a protein, linker protein, which is there. From here, there is one thing which is connecting. This radial spoke will be connecting in the core. This is called as a spoke. In this picture, you are able to see this blue color. This blue color is called as the spoke. Where is it starting from? It is starting from the triplet, and then it is connecting to the center. The center is called as hub. So in between, you are seeing this blue color structure. This blue color structure is your that the spoke. Here you are able to see this red color line. This red color line is the spoke, and this red color circle that you are seeing here is the hub. And here you are able to see that this is it is attached to which part? It is attached to the center is called as a hub. The central region is called as a hub. Okay, there are proteinaceous structures which will connect the central region along with what? Along with the microtubule. And that structure is what we call it as the spoke. Spokes are also connected to AC linkers. The protein linker. Okay. So the final thing is, what is the function of centriole? The function of centriole is, it is the microtubule organizing center. It forms spindle fibers during cell division. in animal cells. Okay, it acts as basal body for flagella. It acts as a basal body for flagella or cilia. Okay, so with this thing we are completing centrioles. Centrioles is over. Okay, so then tomorrow we will see nucleus, and day after tomorrow we'll see nucleus, and we'll complete this chapter. 2 more classes, this chapter will be over. So now coming to the third slide. First 2 slides are empty. Third slide. Fourth slide. Fifth slide. Sixth slide. Yeah, they can act as basal body because they have a 9+0 structure. 7, 8, 9, 10, 11, 12. Okay, with this thing we are completing centrioles. Fifth slide. Yes, both centrioles, both sides centrioles will have 9+0 structure only. Someone wants fourth slide. See. Someone wants seventh slide. See. Someone wants ninth slide. See. Someone wants 11th slide. See. Thank you very much, children. Ninth slide. Okay, thank you. Thanks a lot. Sir, na mudichitte.